I'm Hyobin Jeong from Yonsei University, Department of Systems Biology, and it's my big pleasure to present our research topic in the Sydney Statistical Bioinformatics Seminar Series. So the title of today's presentation is Coupling Single-Cell Genome and Epigenome to Study Functional Consequence of Somatic SVs. So, as Professor Sim introduced, I graduated on postdoc when I studied PhD, and at that time I studied systems biology and bioinformatics to apply these techniques to the disease-related research. And then I moved to Germany for my postdoc until 2022. And then I moved back to Korea and I briefly worked in Hanyang Institute of Bioscience and Biotechnology for 1.5 years. And then um, since 2024, I'm working in Yonsei University and opened a systems biology and bioinformatics laboratory. And our main focus is to analyze bio big data and a multi-layered molecular network, including genome, epigenome, transcriptome, and sometimes proteome as well. So what we want to do is that in integrating these multiple omics layers, we want to know the perturbed signaling network in disease condition. Uh, and recently, thanks to the paradigm shift of single cell technology, now we are able to investigate perturbed network in single cell level. This is particularly helpful when we want to study intratumor heterogeneity. Um, so this is the diagram which shows the clonal evolution in the cancer. So at the beginning, the first cancer cell acquire certain mutation and proliferate and make the clones like this. However, as time goes by, these cells acquire new mutation and becomes another subclone, which is depicted in blue. And this process is continuing through the cancer evolution, and this is very important for the cancer progression and also drug resistance. And this kind of biological phenomena can be more uh, dissected by the single cell omics data and computational analysis. Especially, we are interested in the structural variant, shortly SVs, which is accumulated along the aging and cancer evolution. So structural variant is a type of mutation together with single nucleotide variant, but the size of mutation is relatively large, larger than 50 base pair. It includes copy number alteration or inversion and translocation. And this kind of variant is accumulating during the cancer evolution and interact with epigenome of tumor cells and make the cancer progression. I believe that understanding the interaction between these two layers, genome and epigenome, is very important to know the initiation and also the driving forces of cancer. So to study the two different layers, genome and epigenome, in the cancer, we firstly need to understand the structural variant. This is a key mutational process in cancer, which can lead oncogenic fusion, oncogene amplification, enhancer hijacking, and also sometimes tumor suppressor deletion. And we want to know how SVs affect epigenomic heterogeneity in aging and cancer. To answer this question, we need a single-cell genomics technology which can detect SVs in the single cells. One of the recent methods is Strand-seq. Strand-seq is kind of single-cell DNA sequencing method that resolved individual homologs by sequencing only the template strand of DNA during DNA replication. To do so, we treat BRDU and BRDU to the cell in the culture, and after the cell division, we can obtain strand-specific BRDU labeling, which can specifically remove one strand. And then after the sequencing, we get the read from the template strand. The reason why this is useful is that we can make use of tri-channel processing in integrating read depth, read strand, and phase. For instance, if you don't have any structural variant, you will see the pattern like a reference. But if there's a copy number alteration or inversions, you will see the different pattern in this combination. And this is very useful when we need to deal with the low depth data such as single cell genome, because we don't need to directly sequence the breakpoint, but we make use of the broad area, the pattern of strand state changes. So this is the advantage of strand-seq method. And it will give you chromosome length-wise haplotype aging and also detection of unbalanced and balanced SVs. Using this, we can reconstruct subclonal trees like this. However, we still don't know how those subclones different in their epigenome or functional outcomes. To compare the epigenome of different subclones, we need the technique to measure genome and epigenome from the same cell. As a powerful solution, we attempted to use nucleosome occupancy as a epigenomic readout. The reason why we thought about nucleosome occupancy is that if we look into the Strand-seq protocol closely, there's a MNase digestion involved. And this enzyme is known to cut the nucleosome-free region of the DNA. So after the sequencing, we will get the sequencing read from the nucleosome-occupied region. Therefore, using this sequencing read, we can know the nucleosome position and also the nucleosome occupancy, as you can see in this genome browser tray. And nucleosome occupancy can indirectly inform you the chromatin accessibility in the genome. So using this as a feature set, we can train the machine learning model to infer expressed genes or to infer altered gene activities between subclones. Based on these properties, we devised a computational pipeline scNOVA, which is single-cell nucleosome occupancy and variant analysis. To achieve haplotype-specific nucleosome occupancy as a local effect of SVs, and also to investigate clone-specific nucleosome occupancy as a global effect of SVs. To ultimately understand functional consequences of genetic structural variant. This method I developed when I was doing postdoc in EMBL in Heidelberg. And as a case study, we applied this to the T-cell acute lymphoblastic leukemia patient 1. From the 77 single cells, we could see the subclones emerging by gaining the complex structural variant in the chromosome 6. And using scNOVA, we could find 12 genes whose activity was significantly changed in the clone 2. And those genes were highly enriched with the target genes of MYB transcription factor. And you can see the gene regulatory network here. And there are many interesting axes, including MYB notch-1 axis. And we also validated this hypothesis that when we treat a notch inhibitor to this TALL patient 1 sample, and after some time, the clonal frequency was significantly reduced compared to vehicle control when we treat this notch inhibitor. So by this, we want to suggest that using this single-cell multi-omic analysis scNOVA, we can uncover tumor heterogeneity and also suggest the drug target. And now we are applying this method to different biological questions, and also after I opened the lab in Yonsei University, we tried to continue this application to different biological applications. One of them I want to introduce you briefly is the study of mosaic structural variant in hematopoietic stem and progenitor cells along aging. So during the development and aging, our cells divide very frequently, and this cell division inevitably add more mutations or SVs in our genome. So those mutations could be neutral to the cell phenotype, or it can lead cancer. However, there are many different spectrum in between neutral and cancer, and one of them is clonal hematopoiesis. Clonal hematopoiesis is the clonal expansion of hematopoietic stem cells. In our body, there are a lot of immune cells, and those immune cells are originated from hematopoietic stem cells, and every day these stem cells are dividing and differentiated into many different lineages of immune cells. But if hematopoietic stem cell acquire new somatic mutation and make its clone frequently, we call this clonal hematopoiesis, and this can lead to a higher, and this is linked to a higher risk of cardiovascular mortality and hematologic malignancies. However, so far, clonal hematopoiesis analysis was focused on the point mutations which is occurring in the epigenetic regulators such as DNMT3A or TET2. This is very well-known mutation for clonal hematopoiesis. However, the role of SVs in this clonal hematopoiesis was not fully understood yet. So in this study, we wanted to investigate the landscape of SVs in the clonal hematopoiesis. So we collected the hematopoietic stem and progenitor cells from different ages. In total, 19 donors were participated in this study, and we performed Strand-seq and also the computational analysis from the genome and also epigenome site. The above plot shows the location of structural variant found in these 19 donors, and the bar graph in the below shows the cell type composition. The composition of mosaic SV classes, including deletion, duplication, inversion, and sometimes loss of Y, loss of X chromosome, and a very complex event as well. You can see that the cells from a newborn baby or very young donors didn't show any somatic SVs, but the ages over 60 showed many different somatic SVs, including loss of Y, inversion, and copy number alterations. And using this epigenomic readout from Strand-seq, we wanted to classify the cells into one of the 8 different classes in the hematopoietic stem and progenitor cell lineages. The green one, hematopoietic stem cell, is the most primitive cells which have potential to differentiate any lineages. And there's a multipotent cells, and there's a lymphoid lineage cells in blue, and myeloid lineage cells in red. So we applied scNOVA analysis and built the classifier of cell types. And this shows you the signature genes used for making the cell type classifier. And using this classifier, we inferred the cell type identity of 1,133 single cells from 19 donors. And you can see each of the big bubbles represent one of the 19 donors. And within the big bubble, sometimes you see the smaller bubble when they have somatic SV clones. And within the somatic SV clone, you will see the smaller circles with different colors, and this is the cell type inference from scNOVA analysis. And what we want to do was that comparing the cell type proportion from somatic SV subclone to the wild type control, so that we can ask whether the somatic SV is linked to the cell type bias. For instance, in the right side, you can see that in many cases, the somatic SV show the enrichment of HSC, CMP, or MEP. They were myeloid lineage progenitors, CMP and MEP. So even though the number of donors were quite limited in our study, we could see the tendency that somatic SV clones differentiate more into the myeloid lineage. And then the next question was why mosaic SV clone show cell type bias? So we focused on one of the donor, which showed the bias in the common myeloid progenitors. And in this case, the donor age was 71, and we could see the somatic deletion in the chromosome 17, and some of the cells additionally acquire the somatic deletion in the 17q. So based on this SV profile, we built these clonal evolutionary trees and then compared different subclones using the nucleosome occupancy readout. And this heat map shows the significant genes whose nucleosome occupancy was changed in different clones. Especially the cells acquired 17 deletion showed the aberrant activation of mTOR signaling pathways and also SREBF1 target genes. And we could link this to the genes in the deleted region. For instance, SREBF1 gene itself was deleted in the 17p, and mTOR signaling was regulated by NF1, which is overlap with the breakpoint of chromosome 17q deletion. So we could hypothesize that as the cells acquire this deletion in 17p and q, the SREBF1 regulon was changed, and also mTOR signaling pathway was altered due to the deletion of NF1 promoter and SREBF1 itself. And we wanted to verify this association using the larger cohort in the UK Biobank data. And UK Biobank data provide you the genotype as well as the blood cell count. So we could link the genotype with their cell count in different cell type, including total, WBC, monocyte, and also neutrophil. And it showed that SREBF1 loss can contribute to a cell type bias in leukocyte, specifically toward the monocyte. And among the 19 donors, one more donor I wanted to highlight was the donor whose age was 65. This donor showed the subclonal chromosome X inversion. So as you can imagine, inversion doesn't change the copy number, so the gene copy will remain the same. However, this subclone showed the lineage bias toward the MEP cell type. And we could see that there's epigenomic changes in the SV haplotype compared to wild type. And for instance, the chromatin accessibility of androgen receptor was increased, and the overall AR motif accessibility was increased in the inversion clone. So with this, I want to highlight that using the Strand-seq and scNOVA approach, we can dissect the subclonal structure in the sample, and also we can compare those subclones in terms of cell types and also gene activities. And now I collaborate with Professor Boyeon Park in our department, and also Professor Daewon Jeong in the Hanyang University to apply this method to the cancer initiation in pancreatic cells and also liver somatic mosaicism. And not only this method in our newly opened lab. I want to develop novel computational method and applications to understand the cause and consequence of somatic variant in the genome. And also, my previous research mostly focused on detection of SVs and their functional consequences. But in the future research, I want to also uncover the epigenetic driver of SVs in cancer initiation and evolution. With this, I want to thank to my team members and also my collaborators and my former supervisors. And thank you for your attention.